Hello everyone and welcome to today's webinar hosted by ISEH. My name is Steve Loughran. I'm a postdoc at the Cambridge Stem Cell Institute at the University of Cambridge and I work on how signaling pathways control the differentiation of hematopoietic cells. Today I'm moderating the session titled Changing Concepts in Lineage Commitment, and we have two fantastic speakers today. They will each talk for about 20 minutes and that will be followed by a Q&A session. If you have a question at any time during the presentation, please type it into the webinar chat box and we will address as many of those as we can at the end. So let's get to it. It is my privilege to introduce our first speaker for today, Dr. Marjorie Brand. Marjorie Brand received a PhD in molecular biology from Université Louis Pasteur in Strasbourg, France, where she studied basic mechanisms of transcription. She then did a postdoc with Mark Rudin at the Fred Hutchinson Cancer Center in Seattle, where she used quantitative proteomics to study the role of transcription factors during erythroid differentiation. She started her lab in the Sprott Center for Stem Cell Research in 2004 and is now a professor in the Department of Medicine at the University of Ottawa and senior investigator at the Ottawa Hospital Research Institute. The research in her lab is geared towards deciphering the role of transcription factors in regulating hematopoiesis and understanding how quantitative changes in transcription factors regulate cell fate decisions. And she's going to tell us about some of her work today.
Steve, thank you very much for the introduction, and I would like to thank the committee of the ISEH for the invitation. It's a real pleasure to participate in this webinar and share with you some of our view on lineage commitment. So my lab really became interested in this question through our studies on transcription factors and particularly DNA binding transcription factors. So on this slide here, I'm showing a very simplified model of hematopoietic differentiation with the hematopoietic stem cell on the top, followed by progenitors, precursors, and differentiated cells. Now, hematopoiesis is regulated by a number of transcription factors and cofactors, and these proteins work together as networks to promote differentiation towards specific lineages, such as, for example, the erythroid lineage, which leads to the formation of red blood cells. Now, what's interesting is that the transcription factors that are expressed, that promote erythroid differentiation, such as GATA1 or KLF1, are expressed at very high levels at the specific stage when they are important to drive erythroid differentiation. However, most of these factors are also expressed in a hematopoietic stem cell, although at very low levels, which suggests that the amount or the dosage of transcription factors plays a critical role for cell fate determination. Now, the importance of the dosage of transcription factors for cell fate determination has in fact been established early on by seminal work from Thomas Graf's lab using lineage reprogramming experiments in cell line. And for instance, the lab showed that the first expression of GATA1 in myeloblast cells can give rise to distinct cell fates depending on the amount of GATA1 that is ectopically expressed. For instance, what was shown is that intermediate levels of GATA1 will drive differentiation towards the eosinophilic lineage, while high levels of GATA1 will drive differentiation towards the erythroid cells or megakaryocytes. So this shows that cell fate is directly regulated by the amount of the GATA1 protein. Now, interestingly, it has been proposed that transcription factors that belong to competing hematopoietic lineages, such as KLF1 and FLY1 or GATA1 and PU1, are co-expressed in bipotential progenitors, and that changes in their relative level may be important to regulate cell fate decisions. So the existence of these cross-antagonisms suggests that it might not be so much the importance of GATA1 or KLF1 proteins or their amount that is really important to drive erythroid differentiation, but rather the amount of KLF1 with respect to the amount of its antagonistic partner FLY1. In other words, changes in the stoichiometry or changes in the relative levels between these transcription factors may be what's really important to drive erythroid differentiation. Now, gene regulatory networks, such as this one, for instance, have been established in an attempt to model cell fate decision process in bipotential progenitors. And for example, here is a model describing the cell-fate decision in the MEP by potential progenitor that must decide between an erythroid fate and a megakaryocytic fate. Now, as you can see here, this model predicts that this fate choice is based on competition between GETA1 PU1 and between KLF1 and FLY1. And the idea is that a small change in the relative abundance of this factor may be enough to tilt the balance towards one fate or another. Now, these models or these gene regulatory networks have been designed under the assumption that bipotential progenitors exist as a stable cell state, as shown here in this classical hematopoietic tree of differentiation that I have shown you so far. However, recent work from a number of labs using single-cell RNA sequencing, a technology which has been pioneered by our second speaker today, Allon Klein, have completely transformed our view of hematopoiesis, replacing this classic hematopoietic tree that you can see here into something that looks more like this. Now, in this new continuous model of hematopoiesis, there are no differentiation steps per se, but instead the cells are gradually transitioning along the hematopoietic trajectories. Now, another aspect of this model is that it led some labs to suggest that lineage commitment may occur very early in the hematopoietic stem and progenitor cells compartment, and that cells that emerge from this compartment may be restricted to a single lineage. Now, a major question arising from this continuous model of hematopoiesis is that of the role and the importance of transcription factors for the lineage choice decision. And indeed, as I mentioned, this type of gene regulatory networks were designed based on the assumption that bipotential progenitors exist as a stable cell state, but this assumption is clearly not supported by the recent data on single-cell RNA-seq that shows a continuous progression. So in the context of these new models, the role of transcription factors and really the importance of their quantitative changes for the process of cell-fate decision remains unclear. Now, when thinking about this, one must keep in mind that in these continuous models of hematopoiesis are derived from snapshot measurements of single-cell RNA-seq data. And this approach does have some limitations. For instance, protein levels do not always correlate with mRNA level. And also, these continuous models of hematopoiesis are inferred from cell similarity and pseudo-time reconstruction rather than from temporal measurements that would be important to fully assess the hierarchical relationship between cells. So our objective was to establish a dynamic model of erythropoiesis that could integrate quantitative changes in protein levels of transcription factors over time. Now, standard proteomic approaches such as Western blot or shotgun mass spectrometry are very good at measuring changes in the relative level of a single protein over time. And therefore, we have a pretty good idea of changes in the relative levels of GETA1 or KLF1 during the course of erythroid differentiation. However, these approaches do not provide information on the relative levels between several proteins within the same sample. And therefore, we currently do not know how many molecules of KLF1 are present compared to the number of molecules of FLY1. In other words, the stoichiometry between the cross-antagonistic pairs of transcription factors is unknown at the protein level. Now, if one is to measure stoichiometry or changes in stoichiometry between transcription factors, it is necessary to use methods that provide an absolute quantification. And for this, we decided to use a targeted mass spectrometry approach called selected reaction monitoring, or SRM, which when coupled with the spiking of highly purified isotopically labeled aquapeptides can provide absolute quantification of proteins. And this work was done in collaboration with the lab of Jeff Ranish at the Institute for Systems Biology in Seattle. Now, our model system to study human erythropoiesis is an ex vivo differentiation protocol where the CD34 positive hematopoietic stem and progenitor cells are first isolated from cord blood, and those cells are then differentiated towards the erythroid lineage using a combination of growth factors and cytokines. So to systematically characterize the different phases of erythropoiesis, we harvested cells every second day of differentiation, and we analyzed those cells at the RNA level using RNA-seq and at the protein level using various mass spectrometry approaches. So here I'm showing first the results of the RNA-based data. Here I plotted this RNA-seq-based erythroid trajectory over time along these two principal components, and you can see a nice progression from the HSPC at day 0 that transitioned to the differentiated erythroid cells at day 16. So next, to further characterize erythroid differentiation at the protein level, we used two complementary mass spectrometry-based proteomic platforms. In the first approach, we use an unbiased shotgun mass spectrometry approach, which in combination with ITRAC reagents allows the identification and relative quantification of a large number of proteins. And from these experiments, we identified about 4,000 proteins, 16% of which were transcription factors. And among those candidates, we selected about 100 transcription factors for absolute quantification by targeted SRM. As I mentioned before, targeted SRM allows us to obtain absolute quantification of proteins when it is coupled to the spiking of known amounts of isotopically labeled aquapeptides that are used as internal controls. So to give you an example of this quantification, here is this peptide, this GETA1 peptide, and that is absolutely quantified. And the signal here in blue corresponds to 20 femtomole of this GETA1 peptide that was spiked into our mixture of endogenous peptide. And here in red is the signal produced by the same endogenous GETA1 peptide, and we can obtain absolute quantification of this peptide by measuring the relative area under the curve. And as you can see here, from day 2 to day 10, we see an increase in the endogenous level of this GETA1 peptide, as would be expected for an erythroid transcription factor. Now, looking more globally at the 100 transcription factors that we have quantified during erythropoiesis, on this heat map, you can see that there are several clusters that appear to be temporally co-regulated. Now, one of the questions we were really interested in asking is whether there is a good correlation between protein and mRNA changes during erythropoiesis. And as you can see here, we found that for most of the genes, there is a positive correlation. However, there are also exceptions. So here I'm showing as an example FLY1 and GETA2 genes, for which, as you can see, there is a very good correlation between the RNA level in green and the protein level in red. However, for other genes, such as GETA1 or TAL1, you can see that the protein levels is increasing much faster than the RNA level, which suggests an important contribution of the post-transcriptional regulatory mechanisms. So the fact that we identify these major discrepancies between mRNA and protein levels for factors that are master regulators of hematopoiesis and erythropoiesis suggests that the gene regulatory networks that describe erythroid commitment should not be limited to mRNA, but should really try to integrate proteins. And this is what we attempted to do in collaboration with computational biologist Ted Perkins at the Ottawa Hospital Research Institute. So what they tried to do is to build a network model of erythroid lineage commitments that would integrate quantitative changes in transcription factor, protein, and mRNA abundances. So I'm not going to give any details on modeling, but I will just say a few things. So they are trying to model transcriptional regulation. Proteins are used as an input. They are considered to be regulators. The RNA are used as the output, considered to be the targets. And we can estimate translation rate from the data. So this is a type of model that they were able to obtain from using 15 different DNA binding transcription factors. And you can see we have different models at the multipotent progenitor stage, CMP, MEP, and CFUE. Now, what distinguishes this model from previous model is that it provides an estimate of the relative contributions of all proteins to each regulatory relationships. And we can measure the strength, or we can estimate the strength of these regulatory links. So, and the strength is represented by different level of transparency. So for example, here in the MPP, we have a very strong activation of the FLY1 gene by ERG. However, the strength of this activation significantly decreases when the cells transition from CMP to MEP, and then it completely disappears when the cell reaches the CFUE stage. Now, this also illustrates another aspect of the model, which is that it's a dynamic model, so we can reveal changes in regulatory relationship over time. Now, if we focus on a model in the MEP by potential progenitors, you can see that the model was able to capture the cross-antagonistic relationships between GATA1 P1 and between KLF1 and FLI1. Now, if you look more closely, you can notice that the strength of these different regulatory relationships appear very different, with the repression of PU1 by GATA1 being much stronger than the reciprocal repression of GATA1 by PU1. In other words, this cross-antagonism appears to be quantitatively imbalanced, and we think that this quantitative imbalance is important because it may reflect the fact that the cells are not in a stable state, but instead they may be in the process of moving along the erythroid trajectory. So in that sense, this gene regulatory network is fully compatible with the most recent models of hematopoiesis, with the cells gradually transitioning along the erythroid lineage. And indeed, you can see that when the cells proceed towards erythroid differentiation, the strength of the repression of GATA1 by P1, this link here, gradually decreases until it completely disappears in late CFUEs. Now, importantly, these gradual changes can only be seen because we have integrated quantitative changes in protein levels into this gene regulatory network. So in summary, from this data, it appears that the erythroid lineage commitment is a gradual process, and what we've done here is try to provide quantitative measurements of some of these gene regulatory changes that underlie erythroid lineage commitment. So for the rest of the presentation, I'd like to focus a little bit more closely on the KLF1 FLI1 cross-antagonism at the single cell level. So this cross-antagonism regulates the erythroid versus megakaryocytic cell fate decision. And here I have plotted the copy number of each of its proteins over time. And as you can see, we find that these two proteins are present at equimolar level, corresponding to about 3,000 copies of each per cell in the, at the time where we find the MEP stage. So this data is in very good agreement with the previously proposed model that the switch in the quantity between these factors is important for cell fate decision. However, just with this data, we could not conclude whether the switch occurs in single cell or whether it reflects changes in the proportion of different cell populations, each of which expressing exclusively KLF1 or exclusively FLI1. And it was a very important question to address, especially given the fact that a paper from Tim Schroeder's lab was published suggesting that there is actually no co-expression of antagonist transcription factors in single cell in early hematopoietic progenitors. So to address this question, we decided to use CyTOF because it allows to measure protein expression in single cells. And the CyTOF experiments were done in collaboration with the lab of Doug Higgs, Ed Morrissey, and Chuan Chen at the Weatherall Institute of Molecular Medicine in Oxford. So for the CyTOF experiment, again, we harvested cells at regular intervals during the course of erythroid differentiation. Those fractions were then barcoded separately with palladium isotopes prior to being combined and measured using a cocktail of 27 antibodies, including 11 cell surface markers and 16 transcription factors. And then by using a clustering analysis of the combined 27 markers at the 14 time points, we could identify 18 different clusters. Now, because we have barcoded our sample, we could do a temporal analysis of the cell populations. So as you can see here at day 0, the main population that we see is shown in red. It corresponds to the early multipotent progenitor MPP. At day 2, we see the appearance of the CMP and the bipotential progenitor MEP population. And then from day 4 to day 12, we see the progressive disappearance of these early hematopoietic progenitor cells, and instead we see the appearance of the late precursor cells shown in blue, corresponding to a CFUE and proerythroblast. And finally, from day 14 to day 20, we see the appearance of the basophilic erythroblast, polychromatic erythroblast, and orthochromatic erythroblast, which is the very last stage of erythroid differentiation just prior to enucleation. So the conclusion is that through this unbiased clustering analysis across time point, we could reconstruct the dynamic trajectory of erythropoiesis from the MPP to the orthochromatic erythroblast. So the next thing that we wanted to do was to examine the expression of transcription factors in single cells within these different populations along the erythroid trajectory. And this allowed us to come back to our original question, which is whether there is co-expression of the antagonist transcription factor FLI1 and KLF1 in the bipotential progenitor MEP. And so to address this question, we gated for the EMEP population using the 7 cell surface markers. And when we plot for KLF1 and FLI1 expression in this cell population, we found that the vast majority of the cells do co-express these two antagonist transcription factors. So the conclusion is that yes, there is co-expression of the antagonist factors in the bipotential progenitor population. So next we wanted to determine how the protein levels of FLI1 and KLF1 are changing when the MEP population proceeds towards erythroid differentiation. So here is the expression of KLF1 and FLI1 in the MEP. And when those cells proceed towards erythroid differentiation, we see a gradual increase in KLF1 protein level and a gradual decrease in FLI1 protein level. So these results are very similar to what we have seen by mass spectrometry. So overall, this data is consistent with the previously proposed model because there is a change in the ratio between these competing factors when the cells are differentiating from the bipotential progenitors. However, in contrast to the previously proposed model, we did not see an abrupt switch in these two transcription factors just after MEP. And instead, what we see are gradual changes, and it does take several cell populations for the ratio to be fully resolved. So the next question we wanted to ask was whether changing this ratio might be able to modify cell fate decisions. And to do this, we decided to ectopically express this non-erythroid transcription factor FLI1 in these early progenitors. And for this, we used lentiviral infection to induce the ectopic expression of a flag-tagged version of FLI1. And again, we followed erythroid differentiation using cytoph analysis. So here, to draw this graph, we used the SPRING algorithm, which was established by Allon Klein's lab. And as you can see here on this KNN network graph, there are clearly two trajectories. The first trajectory here that goes to the top corresponds to the erythroid trajectory, as shown here by high level of expression of the alpha globin, for instance. Now, the second trajectory on the left only occurs when we infect the cells with the lentiviruses, and it corresponds to the megakaryocytic trajectory, as shown here by high level of expression of the megakaryocytic marker CD41. Now, what is really interesting with the cytoph experiment is that through the addition of an antibody against the flag tag in our cocktail of antibody, we were able to track and to follow the cells that do express FLI1. And what we found is that only the cells that express FLI1, which are shown here in blue, are differentiating towards the megakaryocytic trajectory, while the cells that do not express FLI1, shown here in green, continue to differentiate along the erythroid trajectory. So that shows that the ectopic expression of FLI1 is able to deviate the erythroid trajectory towards the megakaryocytic lineage. And I would like to just mention that in our ex vivo differentiation system, it contains a lot of cytokines that very strongly push the cells towards the erythroid lineage. But it seems that no matter how much bias or environmental pressure, a change in the transcription factor amount can override these external signals. So just in summary, we think that these results support the concept that temporally regulated quantitative changes in transcription factor protein levels in individual hematopoietic progenitors are key determinants of cell fate decisions in human erythropoiesis. And I have just highlighted here, for the sake of discussion, some points that I think remain unknown and important to establish. First of all, we really do not know the molecular mechanism that underlies these transcription factor cross-antagonisms. Of course, the influence of the in vivo extracellular environment on lineage commitment is also mostly unknown at the molecular level, at least. And we think that future gene regulatory networks should try to integrate additional players such as transcriptional cofactors, for instance, chromatin modifying enzyme, for a more precise modeling of hematopoietic lineage commitment. So I'll stop here and acknowledge people who participated in the work. So I already thanked our collaborators, and from my lab, most of what I showed you was done by a postdoc, Carmen Pali, and computational biologist, Karti Sivaraman. And also I would like to thank our funding sources. And thank you very much for listening, and I'll be happy to take any question. Thanks.
Great, thanks very much, Marjorie. That was fantastic. Yes, just to remind everyone, if you do have a question for Marjorie to answer at the end of the session, please put it in the chat box where the panelists can see them. Right, I'm pleased now to introduce our second speaker today, Dr. Allon Klein. While Allon shares his screen, I'll just give you some quick background about him. So Dr. Klein obtained his PhD in physics with Professor Ben Simons at Cambridge in the UK. He then, for his postdoc, moved to the other Cambridge, doing experimental systems biology with Professor Mark Kirschner at Harvard Medical School. He is currently an assistant professor of systems biology at Harvard, and he studies how cells make fate choices in developing and adult tissues. He pioneered droplet microfluidics for single-cell RNA-seq, the in-drop technique, computational methods for analyzing single-cell data sets, and methods for quantitative clonal analysis. He focuses on the early embryo, epithelial tissues, and the hematopoietic system as model systems to study differentiation. And he's going to tell us about some of his work right now.
Thank you very much. Thanks, Marjorie, also. That was a real pleasure. So I want to, this is actually going to follow up, I think, quite nicely on some of the introduction that Marjorie gave. I'd like to think, invite you to think with me together about the way that we learn about dynamics from single-cell RNA sequencing and about how we can test some of the hypotheses that emerge from looking at these continuum trajectories. And I think the key messages, I'll just summarize that right now, is that single-cell RNA sequencing generates testable hypotheses for the differentiation hierarchy and dynamics of tissues such as hematopoietic tissues, but that if we start to integrate clonal information on single-cell RNA sequencing landscapes, we can start to test some of the hypotheses that are emerging and also look for features that might be hidden from single-cell RNA sequencing or even misleading in single-cell RNA sequencing. And I think that this requires, at this point, fairly little introduction, this idea that if we can take individual cells, measure their transcriptomes, we can start to look for the structure of a hierarchy, order, identify decision points, and identify maybe the mature cell types that emerge in different tissues. And the general paradigm for this can be thought of in analogy to the way that chromosomes can be assembled from shotgun sequencing, where in sequencing we all know that if you have many short reads that are partially overlapping, we can eventually reconstruct a very large chromosome. And in single-cell RNA sequencing, if we take a tissue and we measure the states of cells which are all similar to each other, we can challenge ourselves to assemble a continuum landscape where we put cells which are similar together and eventually get a picture for this. And there's actually now a very large number of attempts to solve this problem. There's a recent paper which reviewed over 75 different trajectory reconstruction algorithms from single-cell RNA sequencing. And on the one hand, that tells you that it's a really well-posed problem, but on the other hand, it also tells you that clearly there's no single answer. And that might also be a reason for concern, which will lead me to what I'll tell you about later. But I'm going to start off by just summarizing some previous work that we've done and also referring to others where you can make some progress just looking at continuum trajectories. So here this is a work that I did together with Mirav Sokolovsky, which was published last year, where we were reconstructing the early hematopoietic hierarchy by single-cell RNA sequencing in the mouse. And a standard experimental design would be to isolate cells from the bone marrow, to enrich for the least differentiated cells so that we don't flood our system with mature cells. Run a single-cell RNA sequencing experiment. Now the technologies for this are quite standardized, and then obtain a large data set from which we can start to look for a structure. And here's an example of representing the structure in a single-cell data set from the bone marrow. You can see this particular representation Marjorie has already introduced. It's known as a spring plot. There's a number of other representations which will do a similar job. And in this representation, each point is a cell, and cells that are similar to each other sit next to each other. And we can see, we can color the cells by gene expression of familiar genes, and we can see that markers of erythroid lineage are in red here. That's the E, define one part of this plot. And then there's the multipotent progenitor sitting in the center, and there's a radiation of states. And as Marjorie pointed out, that what this paints is a picture which looks very much a continuum of states, not a series of discrete balls and sticks. So there's no discrete transitions here. And yet there's a structure to this continuum. It's not completely flat. The multipotent progenitors don't just shoot out into each one of these mature lineages. And if one looks at the structure of this plot and formalizes it using coarse graining techniques, one can reconstruct from here in essentially an unsupervised manner a hierarchy which suggests that there's an erythroid megakaryocyte, but also basophilic and mast cell branch on one side of this tree, and then on the other, then there's the lymphoid cells which emerge very early, and then there's the dendritic cells, monocytes and granulocytes that emerge on the other side. And that this cartoon in the corner is summarizing the bare bones that one obtains by this type of analysis. So just as a comment, this type of analysis is really marker-free, and you can do it essentially in any organism. It's interesting that if you look at a human bone marrow, this is a slightly different isolation procedure. It's looking at Lin-negative cells and actually fractionating them into subsets of Lin-negative cells and then putting it all back together. One sees a very similar topology emerging, where there's the P over here are the progenitor, the multipotent progenitors, and on one side we have a megakaryocyte, basophilic or an erythroid branch, and then on the other side we have the lymphoid cells emerging early, and then again DCs, monocytes, and neutrophils. Very much the same type of picture that we saw in the mouse. And this picture, of course, has every one of these cells has a transcriptome associated with it, and therefore we can take the average gene expression across this tree and start to ask questions about, generate hypotheses. So I'm just going to, a lot of this, many hypotheses that came out of this were tested in our paper last year, and I'm just going to briefly recap on some of them, but I'm not going to spend too much time then showing you the evidence for them. So here's just a quick summary of some of the observations that came out. So in those initial sketches that, or cartoons that Marjorie showed you, basophils and mast cells were firmly associated with granulocytic differentiation, and therefore should be expected to be on the monocyte and neutrophil branch, which would be the green and blue lineages here. In fact, we see them come up on the erythroid side. There's also, so that's one, so there's a fate branch point. We can find the latest point in gene expression at which cells look like they could still move towards a different lineage, and at some point they become committed. The EEPs here are what we putatively called early erythroid progenitors, and the CEPs are putative committed erythroid progenitors. We can now look for surface markers expressed on these cells and identify a FACT strategy to purify these cells. We can look for growth factor receptors expressed on these cells and hypothesize about novel regulators of fate choice. And we can look for interactions between cell cycle and differentiation and identify and hypothesize about those. And in each one of these cases, there's a practical perturbation experiment that can be done in order to test whether the hypotheses emerging from single-cell RNA sequencing are validated. And I'll just show you a couple of those here. So this is looking at novel, or rather unexpected growth factors. We identified 3 growth factor receptors, so the IL-17 receptor alpha chain, RIC, which is a Wnt receptor, and then MST1R, which is a cytokine receptor. And these seem to be localized or at least showing a maximal expression along the erythroid trajectory. And then one can take these cells in vitro, repeating some of the in vitro assays of the type that Marjorie alluded to. We're actually looking at a slightly shorter assay here, and ask about whether the ligands to these receptors will have a functional effect on fate choice. And I'm just showing one example here, which is looking at the number of erythroid colonies, so CFUEs, which form as a function of EPO dosage in the presence or absence of IL-17 ligand. And we can see that IL-17 greatly increases the number of colonies that form. So this is an example of taking an observation, testing a hypothesis, and obtaining some validation that there's a novel role for IL-17 in regulating erythropoiesis. Another example outcome here, which I'll go over quickly, is the idea that we can now use a single-cell RNA sequencing as a way to refine our FACS purification strategy. So once we have identified a candidate FACS strategy from the data, we can go select out subpopulations. Over here, these are the precise FACS gates. Over here are labeled P1 through P5, and then go and ask about then go and purify these cells, do another single-cell RNA sequencing experiment, and ask where do these cells sit, and identify gates which are how pure our gates are. And therefore be sure that when we isolate cells, we're looking at a high purity population with respect to transcriptional profile. And you can see that the P1 gate does a very good job at pulling out these putative committed erythroid progenitors. The P2 gate pulls out this bridge, which are these early erythroid progenitors. The P3 gate was aimed to pull out the early megakaryocyte lineage. And over here, it's instructive to see that although it does a good job at pulling out that little branch, sorry, this is actually the basophil lineage, which is closer to erythroid. Although it does a good job at pulling out this branch, it's not entirely pure. So anybody using such a FACS gate to try to isolate basophilic progenitors should be aware that they may be contaminating their gate with other cell types, and that's very useful to know. And so on, one can do this for P4 and P5. I'll stop there for now. And now, of course, once we have a way of isolating these cells, we can use colony-forming assays to look for the functional, or rather the fate potential of these cells. And we can show using this is work that Mirav's lab did, show that the early erythroid progenitors are the sole population of burst-forming units, or BFUEs, and CEPs are the colony-forming units, or CFUEs, of the bone marrow. All right, so those are two examples. And now I'm going to pivot. So this is, at this point, I'm going to sort of talk about why all of this is problematic. And the reason, and actually Marjorie also alluded to this nicely, is that these cell state landscapes, they generate hypotheses, but they do lead to ambiguities. And some of the ambiguities, anyone who's worked with single cell data will have some appreciation for this, is that there are many choices in data processing which could shape the picture that you see, and therefore the hierarchy that you infer. When we see a branch point, this is a transcriptional phenotypic branch point. It does not necessarily correspond to the decision point of a cell, which could have occurred earlier, or if there's reversibility in state, it could be that the decision hasn't been made yet and the cell can go back. And of course, related to that is that the landscapes show the average dynamics of many cells. And there may be, as Marjorie also described quite in detail, other variables which are not captured by transcriptome at all, such as protein levels, post-translational modifications, epigenetic states, which could have a strong influence on behavior but are simply not present in our data. So these are all of the limitations that come in. So it's great to celebrate the successes, but also worth recalling these limitations. And I'm just going to spell out some of these limitations graphically now. So here's our ideal view of single cell data, which is that we map out a trajectory, we see a bifurcation point, and that the phenotypic point at which we, the phenotypic state at which we see a branch point is also the point at which a commitment occurs. But of course, lineage restriction could occur earlier. That's one discrepancy. We could see a situation where two lineages are indistinguishable, but in fact they're quite distinct from each other and they have different future potentials. We could see a situation where there's an apparent continuum, but it's in fact made of a mixture of two lineages. And it could be that there's a branch point, but in fact only one subset of clones has access to that branch point, but not another, so we have a mixed lineage potential. And there's other ways that things could fail. This is just showing 4 examples of discrepancies that could occur. So what we'd really like to do is to measure the state of a cell and then record its fate. And that, of course, we can't do because we kill the cells. So what we might be able to do is to barcode the cells, let them divide, and then take one sister cell, measure its state, and then record the fate of another sister cell. And for this, we need lineage tracing. So just very briefly, there's now a number of techniques which allow highly multiplex clonal analysis combined with single-cell RNA sequencing. The general challenge is we need to deliver a barcode into different cells. This can be done by integration using lentiviruses or transposases. It can be done by editing a scratch pad and introducing mutations using a CRISPR-Cas9 system. It can be done by random recombination to generate high diversity sequences. And then finally, if we take all that information, the key is that we should be able to read it out using, by encoding the barcode into an expressed transcript so that it can be read out with single-cell RNA sequencing. And there's now a large number of labs who have proposed variations on this theme. And I may not have even captured all their names here on the right. Okay, so I'm going to explore I'm going to show some data from one example, which is a very simple one, so we've decided to call it Larry. And this is just using a single lentivirus, lentivirally encoded barcode, which is just a GFP driven by a strong promoter with a random 20-mer sequence in the 3' UTR. And by infecting cells with this lentivirus after isolating them from the bone marrow in a dish, we can essentially get 3 types of experiment in one. We can look at one sister cell early, one sister cell late. We can randomly sample 2 cells early and then see 2 sister cells early and see how similar they are. Or we can split the cells into 2 different wells or transplant them into 2 different mice and then go and ask about whether lineage commitment has occurred by comparing the fates of the 2 sister cells. So here's an example of some data that emerges in vitro from an experiment of this type. So this is combining 4 time points. This is just the single cell data. There's 130,000 cells here. 50,000 of them belong to clones of at least 1 cell, and there's about 6,000 clones. So we can now start to look for clonal dynamics on a landscape of this type. We can see that the single cell landscape is revealing a multipotent progenitor cell population, which just from the single cell data alone seems to irradiate out into a wide number of lineages. There's a whole set of cell types that emerge, which will be familiar. And now we can start to ask about how clones traverse this landscape in order to identify where commitment points are occurring and so on. So here's just an example of some of the clonal information that we have. We have 6,000 clones. This is just showing 3 examples of clones. On the left-hand side, we have a clone where we detected 1 cell from that clone at day 2, and then many more cells by day 4 and day 6. And we can see that this clone started in a somewhat intermediate state of differentiation, but then bifurcated and gave rise to neutrophils and monocytes, which are the 2 branches that the orange cells are occupying. The middle clone is an example of a clone which self-renewed already considerably by day 2. We can see 4 cells were detected, and then this entire clone moved towards neutrophil fate, every single cell in that clone. And then we see that on the right-hand side, we see a clone where we can also detect cells in all 3 time points, and those cells remained undifferentiated in day 2 and day 4, and then gradually started to differentiate by day 6, but have not moved very far. So just as a comment, we can also do this looking at the cells in vivo if we take the cells out, barcode them, and then put them back into a mouse. And I'm not going to show the clonal data here. This is data which is quite new for us, so I'm just sharing this here to give an appreciation of the richness of the data set. So we have about 180,000 cells. This is transplanted into 6 mice collected at 2 different time points, after 1 week or after 2 weeks post-transplantation. And there's about 4,000 clones that we have now traversing this landscape, and we can really start to pick apart how the differentiation after transplantation occurs and how it differs from our in vitro model. I'm going to focus for the rest of the talk just on the in vitro case, and in fact I've just got a few minutes left. So what I want to do is to really just summarize the types of questions that we can ask, and then just take a deep dive into 1 or 2 of them and refer to a preprint for those who are interested in seeing the rest of the discussion. So with this type of data, we can start to ask, does the hierarchy that we can guess by just looking at the structure of this landscape reflect the clonal, the hierarchy that we see within clones? We can also ask, where are fate commitment points occurring? Are they occurring only at the branch points, and before that clones are essentially multipotential? Or do we see that there's evidence of clonal commitment far before we can see the heterogeneity that's separable in terms of transcriptional states? We can ask whether the clones landing in a particular final lineage all follow the same types of typical trajectories, or whether we can see consistent variation in lineage structure of clones that give rise to the same cell type. So are there multiple trajectories to the same fate? We can ask whether clones that are progressing a time series, say neutrophil differentiation, are consistently moving forward in pseudo-time, or whether when we see cells ordered in transcriptional state, in fact, some of the cells are going backwards, and not all of the cells are consistently going forward. So we can separate between coherent and incoherent flows. And finally, and perhaps maybe most importantly, we can challenge whether the idea that a transcriptional state is indeed a pure state, or whether there's not enough information in transcription alone to determine the state of a cell, and that what we're looking at is a complex composition of committed cells which are just indistinguishable. And this is an interesting question, because one of the big excitements of single-cell RNA sequencing is that we would go beyond FACS to a much higher dimensional picture of cell states, and potentially reach the point where we can take contaminated FACS gates or mixed FACS gates and isolate pure populations. If it turns out that single-cell RNA sequencing itself is not pure, then that would mean that we have more work to do in order to identify a really pure population of cells in terms of its potential and biases in lineage commitment. So I'm just going to focus, unfortunately I don't have time to discuss very much here. I'm going to talk about whether, how do we test whether single-cell RNA sequencing inferred hierarchy reflects the hierarchy of fate choices seen in clones, and I'm going to show you how we can assess where fate commitment points occur. So the first question is a rather simple one. We can now take this picture that we get, formalize the cell state landscape, and generate a hierarchical structure from this. So for example, we would from this picture say that erythroid lineage and megakaryocytic lineages are very close, and then basophilic lineages are somewhat separated from erythroid lineages, but much closer than, say, the neutrophil lineage, and so on. We can make such statements. We can now take the terminal states of these cells, completely ignoring the continuum trajectories, and ask how many barcodes land in each one of these states and which barcodes are shared. And from here, we now get the lineage overlap of different states. And now we can compare how close are cells in lineage to their similarity on the landscape. And when we do this, at least in vitro, we see a very strong relationship, which is reassuring. So the graph connectivity, which is a measure of transcriptional similarity, correlates extremely strongly with the number of shared barcodes. The more similar you are transcriptionally, the more likely you are to share a clonal lineage. So at least in vitro, we can say that it does appear that the state landscape reflects clonal hierarchy, and that's very encouraging. Okay, so now the second question, where do fate decision boundaries lie? And what I'm showing you here is the result of a computational algorithm that just takes transcriptional information and tries to guess the fate commitment bias of cells. And there's now a number of these methods available which will take single-cell data and try to predict where commitment points occur. So red over here would mean that you're strongly committed to neutrophils, blue is strongly committed to monocytes, and then there's white cells are multipotent. So now the question is this is a hypothesis. Is this an accurate description of what is going on? And in order to address this, we can take our data and take every one of our cells that we see early, our day 2 cells, and ask about what their day 4 or 6 sister cells are going to do. And then we can paint our landscape by what the commitment of day 2 cells is. And if we take this and now we just focus on neutrophils and monocytes for simplicity, and we smooth out this picture, what we find is that although the algorithms predict that cell fate decisions are occurring very close to the branch points, because that's really all they can say, we can actually find from the state fate measurements that decisions are committed much earlier. And these cells are emerging from the multipotent state. They're transcriptionally very similar to each other, but already strongly determined. And rather than there being a fate commitment point, there's a fate commitment boundary which proceeds for quite a long way, where cells on one side of the boundary will become neutrophils, on the other side they'll become monocytes, and the multipotent cells can continue to mature for quite a while but still remain multipotent until very close to the boundary. So this is quite a different picture from the one that you would get from computational analysis or just looking at single-cell RNA sequencing data alone. Okay, and of course, once you have this picture, you can go back and ask what genes correlate with these persistent changes across this faint boundary, and we can now start to test the role of specific factors. So this is just an example of one gene which we have not established any causal role in fate decision yet, but we can show that it correlates very strongly with the fate choice that is being made. Okay, so I'm going to stop there. Just to summarize the points that I've made. Lineage tracing establishes the long-term dynamics of cell states and therefore is a useful complement to single-cell RNA sequencing. Using lineage tracing, we've shown an agreement between single-cell RNA sequencing and clonally inferred hematopoietic hierarchical structure, but the fate commitment points of the earlier progenitors is not well characterized by single-cell RNA sequencing, and it appears much earlier than computational algorithms on existing single-cell RNA sequencing data would give you. And I also highlighted several other questions, and I will, due to lack of time, just refer you to to our preprint and hopefully sometime soon to a publication in which we address the rest of the questions. Just to give some credit, so the early work just characterizing the hematopoietic hierarchy in the bone marrow was done in collaboration with Mirav Sokolovsky's lab, and that work was done by Sam Wallach in my lab. And then the later work was a very strong collaboration between Caleb in my lab and then Alejo, who's a fantastic postdoc in Fernando Camargo's lab. And that was this clonal analysis that I just told you about. So thank you very much.
Thanks very much. Fantastic. So it's time for the question and answer section. So I might just kick off then while we wait for the attendees to ask a question. I had a question for Marjorie about what the transcriptional consequences are for a transcription factor gradually changing in its own expression. So if a transcription factor gradually increases, does that mean all its target genes also gradually increase, or is it more complicated than that?
I mean, of course, we would expect that the target genes for these transcription factors, some of them might gradually increase, but you could have a lot of different situations because these transcription factors might progressively replace other transcription factors that are bound to the same target genes, in which case you could see an on-off state as well for some of these target genes. Excellent.
Thanks very much, Marjorie.
I have received one question. So the question is there any reason to use a transcribed barcode? So there really are two ways. That's a good question, and there really are two ways to do high diversity lineage analysis. One of them is to just create a DNA barcode which is non-transcribed, and the other one is to make sure that it is transcribed. And if one uses a non-transcribed barcode, one can still get very far by fact sorting out defined populations of cells and asking which barcodes are present in which isolated population. That gets you so far, but it does require that you have a good way of isolating each and every one of the populations of interest in advance. So the advantage of using a transcribed barcode is that you can, without any prior knowledge of FACS gates or the structure of a population, directly associate the presence of a barcode with a transcriptional state as seen by single-cell RNA sequencing. So all of the data that I showed you would have been impossible to access if the barcode wasn't transcribed, unless one uses a clever method for accessing DNA in parallel to RNA in single cells. And at the moment, with current technologies, that's much harder to do. So, so with current technologies, just having the barcode express provides a very quick and efficient way of accessing clonal information with transcriptional information together. Hopefully that answers the question.
Excellent. Well, I might also abuse my privilege as moderator to just ask another quick one. And this is to both of you, really. I don't have a good impression so far from the data of how similar the dynamics of differentiation is across cells. So for example, going from a stem cell to an erythroid cell, does every cell do that taking about the same amount of time, or makes the same number of divisions, or can that be wildly different between individual stem cells?
I guess I could start. I mean, just to say, we really don't know. And in the in vitro system, we do have cells that accumulate at specific time or specific region along the trajectory. So I would expect that you would have some points where the cells may be blocked, and that might be not blocked, but that would points of amplifying, and that may be related to a cell cycle, I would think. But yeah, in vivo, I think it's a very hard question to really address.
Great. That completes sort of the hour of the webinar, and I'm not seeing any outstanding questions. So we might close the session there. So it just remains for me to thank our two speakers today for their fantastic talks. I'd also like to thank Tamara White from ICH for helping with the organisation of this webinar. But with that housekeeping, just remains for me to thank you all for attending and to thank the speakers.